Spermatogenesis is the process of germ cell differentiation that leads to the development of spermatozoa. The normal physiology of the hypothalamic, pituitary, and gonadal axis and how it contributes to the formation of spermatozoa will be reviewed. Here is a cross-section of the brain. We will now focus on the hypothalamic pituitary axis. The hypothalamus and the pituitary gland are linked by the portal-vascular system. The portal-vascular system delivers hypothalamic hormones directly to the anterior pituitary and avoids the systemic circulation. Let's zoom into the hypothalamus. Of all the hypothalamic hormones that work on the pituitary gland, the most important one for the male reproductive system is gonadotropin-releasing hormone, or GnRH. In the hypothalamus, the neurosecretory cells synthesize GnRH, which is secreted in pulses. Into the hypophyseal portal vessels. The pulsatile release of GnRH prevents downregulation of its receptors in the anterior pituitary. There, GnRH stimulates the gonadotropes of the anterior pituitary, which then produce a pulsatile release of luteinizing hormone, or LH, and follicle-stimulating hormone, or FSH. Let's zoom into the anterior pituitary. LH and FSH travel from the brain to the testes through the blood vessels. Let's look more closely at the cross-section of the testes, and even further to a section of a seminiferous tubule. FSH binds to Sertoli cells and LH to Leydig cells. LH stimulates Leydig cells to produce testosterone through the use of cyclic AMP or protein kinase as a second messenger. FSH stimulates Sertoli cells to produce androgen binding protein, or ABP, also through cyclic AMP or protein kinase as a second messenger. Testosterone diffuses into adjacent Sertoli cells where it is required for normal Sertoli cell function. ABP binds testosterone to help maintain a high local concentration of testosterone within the testes. This promotes the development of germinal cells during the process of spermatogenesis. Let's take a closer look. Testosterone separates from androgen-binding proteins and moves through the cell walls into the cytoplasm of Sertoli cells. It then travels to androgen receptors, which causes the dissociation of androgen receptors from heat shock proteins. Testosterone binds to androgen receptors, which leads to dimerization and phosphorylation. The activated testosterone, or androgen receptor complex, then moves into the nucleus and interacts with androgen response elements in the promoter regions of target genes. Co-activators such as ARA70 also bind to the complex. This allows it to interact more easily with the general transcription apparatus, GTA. The activation of target genes leads to increase in the synthesis of proteins and growth factors, thus promoting spermatogenesis. Now that growth factors have been produced, the process of spermatogenesis begins. Spermatogonia first proliferate by mitosis to become diploid primary spermatocytes, or 2N. Primary spermatocytes are then transformed into 2 secondary spermatocytes, or 1N, during meiosis I. Each of these cells, in turn, is converted into 2 1N spermatids during meiosis II. These migrate towards the lumen of the seminiferous tubule. During spermiogenesis, Spermatids undergo a dramatic change in form. They evolve from round cells into stem-like spermatozoa, which are ready for fertilization. To regulate this process, Sertoli cells, under the influence of FSH, secrete inhibin. Together with testosterone, inhibin travels through the blood vessels back to the anterior pituitary. Inhibin provides negative feedback for FSH. Testosterone provides negative feedback for LH at the level of the anterior pituitary and also provides negative feedback for GnRH in the hypothalamus.